Okay, so we've installed the software. Now let's solve the PSP28 structure, starting with integrating the data. So let's make a directory called solve. Integrate and XDS. Okay, let's look at the data. Okay, so this is a directory that comes from Diamond of CBF files that contain a PSP28 dataset. Let's look at the log file. Nothing. Okay. Let's look at the headers of one of these files. So this is a CBF file, which is crystallographic binary file. It comes from a Pilatus detector, which is a nice pixel detector. These data were collected at I24 at Diamond in 2012 on this MX7299, which is the Imperial Data Collection Group, and it was crystal A32, and it was a square rod crystal. The exposure time was 0.2 seconds. The pixel size was 172 microns. The wavelength, which we will need to know later but should be in our data when we process it, is 0.9778 angstroms. We need to know the beam center in pixels, which our integration software will pick up. We started at 71 degrees and we rotated by 0.15 degrees each image, and then we're into the binary data. So we had 900 images, so the degree of rotation that we had was 0.15 degrees times 900, which is 135 degrees of crystal rotation. So we can look at these images. The data set will be relatively okay. So this shows the ADXV shows the image. Autoscaling scales the whole image to the window, and the high spatial frequencies of spots often don't survive this scaling. So I look at the image at 100% scale, which is one screen pixel is one image pixel, or 50%, which is two screen pixels is two image pixels. So we can see we have a reasonably nice diffraction pattern. Some of these spots look a little bit split, but it looks perfectly okay. If we draw a line through the spots, we can see each spot, and it's predicting a lattice, but it's confused because it's looking at this boundary. So let's look at another image. The original test image has more spots on it because it's a larger rotation. So if I draw a line through these spots are close together, which means they correspond to a large unit cell. That's a unit cell of about 180 angstroms. And in the other direction, the unit cell is, the spot spacing is larger, which means the unit cell is smaller. So just drawing a line through those, it comes out at 56 or 57 angstroms. So when we integrate this data, we should be looking at a unit cell of about 180 by 56 angstroms. The other thing to note is that the crystal is mounted with a rotation axis I think horizontal, and which means it's rotated around this this axis, which is the correct orientation, because the long cell axis should be slightly offset to the crystal rotation axis, which it is. It's along the horizontal of this image. If it was vertical, we might have problems with spot overlaps. And this looks like nice diffraction. if we look at this image, the spots, let's magnify, the spots are going to about, I can see a spot there at 2.4 angstroms. The rule of thumb is that the spots, the resolution typically goes a little bit further than you can see. The other thing is how you see, you need to adjust the scale, because a typical screen has only about 1,000 levels of grey, whereas this image is recorded probably 16 bits, which is 32,000 or 64,000 levels of grey. So the structure in the image is more than you can actually see, so you need to adjust the scale to see, say, a spot here better. Okay, so this is the directory that has the images in, so I'll just take a note of that. And then let's go to PSP28 solve and our integration directory. So we're going to use Xia2 to integrate. Let's look at the options. I need to give it this directory and it will automatically extract the images, which are the data sets. Here we go. Okay, so I'll make a comment and then I'll build up a command line path ending in the directory. Okay, so I'm going to use 3D2 to integrate, which is XDS using all images for indexing. nproc. I have 4 processors, but I'm only going to use 2 of them because I need others to run the video. I don't know the space group, I don't know the unit cell. It will take the beam center from the images. I want to do anomalous diffraction processing. The atom, the standard anomalous atom in proteins is sulfur, so I'll pick that. Project, this makes the files have nice names, and puts information into the files. It's PSP28, and the crystal is A32. And okay, so that's my command line. It seems okay, so I will now uncomment it and run, and this will take about 20 minutes.